indigenous IMOs or indigenous microorganisms on my farm, which I guess are driven by the monocultures that I also employ and some of the sort of limited rotations on my farm as well. So it has been a good journey that's seen me sort of work to a farming system that's certainly less dependent, almost not dependent on fungicides and insecticides, but every now and then I do need to intervene. And I guess the biostimulant that I've used has just been a blend of up to 40 or 50 plant exudates. So it's sort of been diversity in a bottle. And that has enabled me to sort of really, I guess, see a very strong microbiome underneath my crops. And from that, I've seen a lot of improved nutrient efficiency, obviously disease suppression. But obviously, that has taken me so far, and I guess back in 2015, I did my initial testing on taking some samples from the rhizosphere and sending them away, and got back a nice cluster graph and all this sort of stuff. But apart from getting some data, it didn't really mean a lot to me. And I've been presenting for the last 14 years at the soil biology masterclass that has been held at the University of Western Sydney. We'll be in Western Australia this year. Professor Brudge has just moved over to Western Australia, so that might be something for you that are interested in soil biology to perhaps attend that. That'll be in September. But Peter and Ben actually were at that course in 2024, and they introduced me to some of the work they were doing around sort of the greater and growing understanding of what functionality different parts of the soil microbiome play in our farming systems. And I guess that was really exciting because it gave me the opportunity to actually revisit testing some of my soils to see what biology I had present, and then to see how that functionality was, and hopefully over time identify maybe some biological sort of families that weren't present in my soil that could hopefully take it to a different level. So we did some first testing in 2024 on a couple of paddocks, a heavy site and a lighter site. And then we did some backup testing last year, and we also introduced some biology that was sort of seen as perhaps missing in my soil last year at a first step. And it's an ongoing journey of discovery and learning. But what it did provide me with, which was really quite exciting, was some science, I guess, that backed up some of the observations that I was making on my farm on having, some pretty good resilience to sort of pathogens and insects on my farm, and some of the microbiome that was responsible for providing that were quite present in the testing that we did, but there were some missing gaps as well. So we're still learning. But what's really exciting for me about this is that, and Ben and Peter will explain it much better with some good examples going forward, is that I think once we understand what our total nutrients and minerals are in our soil, then we can marry that to see if the microbiome's present to make those nutrients available for our crop. And of course, it's much bigger than that, because we know that as a crop that's or pastures, because I do run cattle as well, that are supported by a healthy microbiome, also have a lot of other compounds in there that are produced to sort of build that resilience to sort of warding off insects and pathogens and funguses and bacteria as well.
Developed. It actually
Someone's got their speaker on.
Analysis for you. So if you have herbage samples.
Don't know where that's come from. That's all right.
Yeah,
I'll carry on.
Everyone.
So really exciting, because I think we're reaching, I feel as if we're potentially, with this type of functional understanding of what we have present, we will be able to identify potentially what organisms we can actually put into our soil, because there's a lot of people out there selling bugs, but I personally believe we can do a lot of work restoring a lot of our dormant biology in our soil, so that we have some a pretty good healthy environment that we are introducing potentially new biology to, where it's got greater success of surviving. But we can, if we sort of start cross-checking, what nutrients we have in our soil, and if they're not becoming available, then it may well be that we just don't have the P or K or solubilizing microbiome in our soil. So to me, it's an exciting way for us that are working in this movement back to working with more nature. We can really start delving in scientifically to what's going on. So I might leave it there, Carrie, and hopefully we've got plenty of time for questions later on, because it is going to be very context specific on, your environment and your farm, on sort of how you work this out. That's the whole idea. So what I do here is it's been a long journey of different changes. So I'll hand it back to you, and then you can pass it on to Peter or Ben.
All right. Thanks for giving that context, Stu. I think that when I started this webinar, I gave all the introductions and then I started the meeting. So if anyone felt like they were missing a little bit of context up top, that was my bad. So I might have to do my introductions again. Am I correct in thinking that everyone missed that portion? Yeah, awesome, great. Okay, so Stuart McAlpine, who you just heard from, is a Buntine farmer. He is one of 5 farmers that is participating in Regen WA's Natural Capital Accounting project. And then we've got Peter and Ben online joining us from Greenmate Agriculture. They come into the project because one of the new metrics that we're adding when we're revisiting these 5 farms is soil functionality and soil biology. So yeah, today we're chatting with Stuart about how this all factors into his farming methods, and then Peter and Ben are going to talk us through the methodology behind the soil functionality testing. So yeah, if anyone has questions through any of this, just drop it into the Q&A box and we'll have plenty of time at the end for people to ask them as well. I'll take you all off mute and we can have a proper conversation. All right, hopefully that clarifies things. Are you able to share your screen with us, Ben? You've got slides.
Yeah, let's give it a go.
So while Ben's doing that, I'll just quickly introduce us and Ben. Again, Pete Briscoe, Greenmate Ag. We've been around for around 5-6 years now, and Ben's been with me nearly the whole time. Personally, been in the biological movement for over 20 years now. Got to live in the States, saw some real progress over in America with this, and met a company over there called Biomakers that we're the sole distributor of in Australia. And I was very excited to bring this technology back to Australia on the basis of adding and filling in missing data that we're always trying to chase when we're making decisions for farming. And I think probably, this comes from me being a company that sells products as well as farming services. We are too product driven in this industry. We're always looking for something that's going to fix a problem, mask a problem, silver bullet, usually something we hear a lot about. But it's a very complex system. And usually what we're finding is you're not getting enough data to make those decisions. We're only looking, if you look at that triangle of chemical, physical, and biological, the biological component of what we're always doing is usually just completely overlooked or glossed over, with most focus on the chemical, and then physical would be next. But when you understand how plants want to function, they are the hierarchy in the soil, and they are constantly signaling into that soil structure, asking for different things at different times through electromagnetic signaling, and calling different microbes up into the rhizosphere, the root system, to perform various functions. And they're not just all around nutrients. They can be around protection from biotic or abiotic stresses. So, extreme heat, extreme cold, lack of water, pest, disease attack. And even that the nutrient cycling side of things, I think a lot of us sometimes forget that when you put a fertilizer into the ground, it has to go through biological processes to then actually get up into that plant. So having those required microbes that the plant's looking for at all times in that system is critical for the end result that you're looking for at the end of whatever season you've done, and what results and outcome you're looking for. So what Ben's going to run you through now is the test that we send off to the States. We're actually currently in talks with a lab here in Australia, in Victoria, called ALSA in Mildura, to bring this testing to Australia so we can actually do the sequencing here without having to send the test over to the States for sequencing, because it does take around 4 to 6 weeks to get the results from this, based on having to send and ship the samples over, sequenced in California, and then they run it through their matrix to get the end results. But what Ben will run you through, and what excited me the most about these tests, is a lot of tests we seem to do are data on a bit of paper. And half the time, even we look at things, it's very difficult to understand what that data is actually telling you and how to correlate that data together to then realistically come up with a end process that you're then going to proceed forward with, because that's what it comes down to, coming up with something at the end of looking at all this data to decide, what am I now going to do next, whether that's an input or something you're going to do on the farm. So what this data is able to give us is a good snapshot and a highlight of what's going on biologically, but it breaks it down into 3 different sections, which Ben's going to run you through. What the first section being pathogens that are present in your system, and any microbes that are there to be able to suppress those pathogens or keep them at bay from attacking the plant. It also looks at what pathogens are not in the system, which is critically important as well, because I find a lot of the time we seem to be putting chemicals out and treating things that we don't necessarily have. The second part of the test looks at stress adaptability and the hormone production that's going on for the plant to be able to withstand those different abiotic and biotic stresses. And finally, looks at nutrient cycling and the microbes that are in there solubilizing and converting nutrients into a plant available form, but also what they're competing with and where you're seeing gains and losses in your soil system. So over to you, Ben. I hope that gives a bit of an overview of what this part of it does. And what we do is then take the data from this and the data from the other tests we do, whether that's standard soil testing, water, all the other things we do on the farm, historical testing as well, and then put that into a report that's easily to digest for the farmer, so that you can actually look at a high level, here's everything that's happening in your soil and everything that's happening on your farm, and here's what we recommend as far as a path forward to get the best bang for your buck. And it is a bit of a journey. I do find at times we try to do too many things at once, and you can spend a lot of money and not necessarily get the end result, because you can get those counterproductive things happening within the soil when you're doing too many things at once. So it is a bit of an understanding of, well, what are the best things for me to do next? What is my budget? That's critical as well. And how do I get my farm moving forward year by year? So over to you, Ben.
No worries. Thanks, Pete. So I'm sharing my whole screen just because I want to look at some things in the browser. So can everyone see what I'm sharing on the screen now?
Yep.
Yeah, cool. So the first thing I want to go through is the sort of summary report that comes or sort of automatically generated when the DNA test comes back. So the way that we do it the same as a standard soil test, usually at the same time. I'm going to take a composite sample over whatever designated soil area we're looking at, and then take that from the same bucket as our standard soil test, so we can get a real one-to-one comparison of the chemical aspect of the soil with the biological aspect as well. So that we send off to a company in the States called Biomakers, and they run this analysis, which is a proprietary machine learning algorithm. It's been going for quite a few years now, so it keeps getting better and better. What it does is it takes the DNA and the RNA in the soil, it processes that all out, it identifies what are the known organisms and assigns them to a function. And then the machine learning part of that, it takes all the unknown organisms, which tends to be most of the soil biome, and then associates them to the closest known organism and infers their function, so that we can get a really comprehensive map of the functionality of our soil microbiome. So as an overview, we get this generalized soil biosustainability score. That's a very high level sort of thing. I think it's more valuable to sort of dig into the details of what that's telling us. The front page here just has a few sort of highlights of major areas of improvement. So going down, there's some pretty standard stuff for soil microbiological tests. We've got total counts of bacteria, fungi. We've got a ratio of arbuscular, mycorrhiza, fungi, and then our fungal bacterial ratio, and then sort of just another overview that's going to get expanded on as we go down. Here, as sort of Peter mentioned, we've got our health of the soil. So this is all of our biotic stress. So we've got all the diseases that have been detected, and then a risk rating based on their abundance, the types of plant, the crop that we're looking at that's in the soil, and the risk of that particular pathogen to the plant, as well as everything that's not detected. So we don't need to be assuming that we have a pathogen in the soil when we don't necessarily have that. We also then look at specifically what's in the soil that is controlling that in a biologically organic way. So whether that's predation, competition, suppression, imitation, all these different aspects of different biology that can control pathogens in the soil, that's sort of split up and rated as well. So here, if we're looking at largely fungal or can be treated as fungal disease, the most relevant biocontrol agent there is going to be fungicidal control, and that's relatively low. So definitely a big area of improvement there. Going down again, now we have that sort of sympathetic production of plant growth hormones that happens in the rhizosphere to complement what the plant can produce by itself. So this soil in particular is doing quite well. So that plant can really have that benefit of having additional growth hormone produced when maybe conditions aren't quite right above the ground to help the plant grow. Stress adaptation. So the terminology we use is resistance and resilience. So resistance being the plant's resistance to biotic stress, whereas resilience is our abiotic stress. So here we have all the compounds and things that are produced in the rhizosphere to help the plant deal with things like drought stress, cold, heat, wind, heavy metals, salt. So all that's sort of broken up, and something we would generally see is that as a system with limited resources, if your soil doesn't have high levels of heavy metal, we wouldn't see a high level of heavy metal resistance because it's just not something that's needed. And the system, being sort of smarter than we historically give it credit for, is going to put those resources where it's needed. So you might see a much higher investment in the soil biomes adaptation to salt tolerance or iron tolerance, for example. Again, sort of looking at nutrition and carbon. So we get an overall rating for these elements in the soil, and that's the first column here is a general rating, but then we break it down into the first column being this one, sorry. Then we break it down into gains and losses. So what species in the soil are interacting with the carbon cycle in a positive way, and what are interacting with it in a negative way for what we want to achieve. So with carbon, we're looking at what's helping root exudation and free fixing carbon be stored permanently in the soil, whereas with loss, we're looking at what's removing it through fermentation, methane formation, respiration. And then we can also see how healthy is the actual breakdown, so the decomposition pathway of getting green matter back into more permanent soil carbon. We assess NPK the same way. So nitrogen in particular is very, very heavily affected by different soil conditions and the biology that are influenced by them. So if we've got, low levels of nitrogen release, which is the supply of nitrogen to the plant, but then it's being consumed at a higher rate than we would like by organisms in competition with plants, that's definitely something we want to adjust to make the soil a lot more efficient in the way that it cycles nitrogen. We then sort of look at the same, a bit more brief with the microelements, so we can marry that up to our soil tests and look at what are our issues in terms of total nutrition, and then what are our issues in terms of how that nutrient is being processed in the soil. So that's a quick overview of the summary. I did want to run through a couple of other things on the portal. So all the information we get is stored and processed on an online portal that's specific to each farm. So that information stays there forever, which lets us sort of track things over time, really easily compare different parts of the land for trials. So here, this is just a bit of a fun visual of the microbiome of a specific farm. If we're interested, say, in a specific organism, we can pick that up, it'll highlight it, and then I've got a count. So whatever it is in cell count, and then as well as a percentage of the microbiome. We do it as both because we want to be able to see how a certain organism tracks over time with seasonal variations. So a total cell count is not necessarily as useful looking at a very wet summer to a very dry summer, because the scope of the entire microbiome is going to expand and contract with seasonal conditions. But if we're looking at a percentage as well, or we have the ability to look at it as a percentage as well, we can sort of track the effect if we're looking at something beneficial or less than beneficial. Like if we've got all our pythium here is 11%, we definitely want to see that coming down. Here we have that sort of comparison tool I sort of touched on. So if we've got 1, 2, or 3 samples, we can do that 1, 2, 3 paddocks, 2 trials, and a control, and like look at a specific species that's an indicator of disease or disease control, nitrogen fixation, carbon cycling, and compare our inputs or our practices effects on different trials across 1 year. Or we can look at year 1, 2, 3 of a paddock that we're treating to sort of see how that's having an effect over time. This one's just a pretty simple Venn diagram. We can look at a few metrics around similarity and overlap between a couple of soils, 2 to 3. This is just an example of the value that we can sort of get when we look at comparing the standard soil information to the biological stuff. So I like this example because it shows this is from a turf field, so it's very sandy. The manager's been putting out quite a bit of potassium, so there's more potassium than the plants can use, and you'd think that's a lot of K in the system. That's great, it'll draw it down. But when we look at the biological aspect of the soil, what's happening is it's rated really, really low. So all that available potassium is bypassing the natural K cycle, and all of those organisms that are supposed to push that pathway and provide potassium to the plant are all suffering. They're bypassed, they're not needed, they're being antagonized just by not being used. And then all of the organisms in the soil that are consuming potassium in competition with the plants are feasting. So they're being fed, they're being promoted over the ones that we'd actually want to make the soil a lot more efficient. So, it's a good example of where you can really highlight maybe the fallacy of having a lot of nutrient where you don't need it. Yeah, I'd say that's probably a decent overview. I might leave it there just for time.
Thanks, Ben. Carrie, someone asked a question if I'm able to answer.
Yeah, we can get into the Q&As.
Yeah, someone popped up in the box around scientific papers. I'll have to go back to Biomakers on this, but what really stimulated us was mid last year. They're running a program with a biostimulant company actually in the States, and they're doing a study with some government funding on looking and checking through the microbiome in the Midwest of the US in cotton and corn fields. And a lot of these fields that they were looking at are using, 1 year they'll do some corn, next year they'll do some cotton. So they wanted to see when they put their biostimulant out and different variations in years and seasonality in weather patterns, how the soil microbiome functioned and what was becoming more prevalent during different cropping cycles, which I found absolutely fascinating. And again, going back to that comment I made earlier around the plant being the hierarchy in the system, we need to understand that the plant is continually the one that is bringing to the fore or suppressing the different microbes in the soil structure based on what it is looking for at any specific time and period of its growth cycle, as well as different plants requiring different microbes. And what they found was that when they were putting their biostimulant out, the plant was using that the biostimulant was obviously creating more bodies in the soil structure. The plant was bringing different microbes to the forefront during different cropping cycles and different seasons. It was actually absolutely fascinating. I'll have to find out if that's been written up into a paper. But if there's anyone on here that's there's any government funding around, I'd love to be looking at doing something like this in different areas, because it's a very powerful tool for you to then again, over a period of time, understand what drivers you need to do when you're putting a particular crop in based on the type of season you're getting as well, and allowing that plant to function the way it wants to function. this is half of the issue we have in agriculture is we're constantly, I remember a soil scientist telling me one day that stuck with me forever, this was going back 15 plus years ago, that we need to get the foot off the plant's throat. We're constantly putting things onto the soil and the plant, thinking we're doing the right thing, but we're actually, a lot of the time, inhibiting the function of how the plant wants to behave and how it wants to perform. And the plant itself, as I said, is controlling what's going on. We've even discovered in the last couple of years that plants' exudation not only attracts and stimulates certain microbes to come up to that rhizosphere root area, they're also distracting and dissuading others to go away. So plants are quite powerful in how they utilize their exudation. And we're finding a lot, it comes back down to the plant's ability to photosynthesize and create energy for itself, do these different functions. And energy is the key. Looking at the way a plant can get that photosynthesis up to a really high level gives it the ability to have more energy reserves to do a range of other functions. Again, just a little tidbit, I'm not sure you're aware, but when a plant is given nitrogen in nitrate form, it has to take that nitrogen internally and use photosynthetic energy to convert it to firstly an amino acid, and then it converts that amino to a peptide, and then a peptide is then converted to a protein, and the protein is what provides cell elongation within the plant. So this, again, all of these different functions are using this photosynthetic energy. It's a finite resource that the plant has. So if a plant doesn't have the energy requirements to do certain functions, they just skip them. And the beauty of what these soil microbes do and nitrogen fixing microbes, there's a range of different things that nitrogen fixing microbes do, and they're not all created equal. Certain microbes in the soil structure in nature can take N2, which is 73.5% of our atmosphere, by the way, which falls to the soil and percolates in the soil structure. It's two nitrogen molecules bonded together with a weak magnetic force, and a particular set of microbes take that N2 and consume it. And what they then spit out is ammonia. So then that attracts ammonia microbes to come along and eat that ammonia, and then they convert that to nitrite. And then the next level of conversion going on in the soil is nitrite conversion with another set of microbes into nitrate, which is a form that the plant can easily take up. Now, there's even been discoveries to show that at that rhizospheric level, certain microbes can start that conversion from nitrate to amino acid, allowing the plant to have more energy reserves for when it starts converting it internally. So we are still on the precipice of not understanding how this works. I mean, if you ask any quality soil biologist, they'll tell you if we know 5%, we're probably overstating it. So this is all new learning for us, and it's tools like this that give us a better understanding of being able to, like I like to say, pull the hood up on your car and actually see how the engine's functioning, and then allow you to understand how those functions are interacting on your little patch of land. And this is the beauty of doing these things for your patch of land, because it's farming seems to be a lot where we like looking over the fence. What's the neighbour doing? What did my dad do? What did my granddad do? And these are things we sort of get stuck into. And we do these processes without much thought on the effect that it's having on the soil microbiome year to year. So I'll see if I can find something from that. Whoever put their hand up on that, and we'll send it over to the guys at Regen WA if there is a paper on that. But it was I might even be able to find the link to that that webinar. It was, again, about 45 minutes, and Ben and I walked out of that webinar absolutely blown away by the differences that were happening in that microbiome over a number of years, and the crop type on the same patches of soil using the same ingredients. Completely different results. So, and I think I just had a conversation with Stuart earlier this morning, and, again, as Stuart mentioned, we're up against it because a lot of the time we're monoculturing. So that, again, is nature doesn't work that way. Another one of my soil biologists that I like talking to a lot, one of his famous comments he says to me is that nature doesn't want a field of plants hanging out with all their friends. They want diversity. They want differences out there. They want different grass types and different species, so that soil microbiome at each of these different plants and trees are calling into there. They're attracting different sets of species for the jobs they need, and it creates this major biodiversity within your soil. The key is to have those microbes there for the plant or tree to get the response when they call. So I hope that answered that question. Sorry, Carrie, I just wanted to jump in while someone put their hand up while it was fresh.
Yeah, no, that's great.
Back
Thanks.
over to you.
I think, yeah, we were going to jump into the questions anyway. And there's one here that I'd like to expand on and add a kind of secondary question to. There's a question from Greg about whether the planting of multi-species cover crops reduces the need to introduce soil microflora. And I suppose off the back of that, I'm wondering Obviously, the soil testing that you do generates a lot of comprehensive information, but how much information are you also collecting about management practices on farm? Or Stuart, how much can you extrapolate from the programs and the reports that you're given about how your management practices might have been influencing these results, or what can be tweaked beyond different inputs or products to move things in the direction that you're aiming for?
Do you want to take that one to start with, Stuart? Because you're the man on the ground, been doing this for a lot longer than I have too, directly.
Yeah. Look, it probably still getting our head around this, all of us, including, Pete and Ben, and just trying to understand what it all means. And I guess in the absence of sort of getting more support from our research institutions here in Australia, because obviously our context is a lot different, more ancient soils and sort of heat stresses. But I think what's really exciting about it is it's not until we probably do more testing on, different parts of the farm, different seasons, comparing multi-species pastures to monocultures, that we'll probably learn a lot more about, the potential increased capacity of our soils, because we definitely will be lacking stuff because we're not growing indigenous crops and species generally anyway. So there will be stuff that will be beneficial. So, but I think for me, just starting to track what's going on in different seasons and different crops. Like, we've only done 2 seasons and they've both been cereal crops. One of those paddocks will be a canola crop this year. Another is a lupin crop. So it'll be interesting to see, the difference that those crops have on the DNA testing that we'll do again this year. And then we applied some different biology last year and some strips. So it'll be interesting to see whether some of that starts expressing itself, into subsequent seasons as well. But again, I think knowledge gives us the ability to educate ourselves and learn more. And I think we are yet to identify or name a lot of the microbiome in our soil, like Peter articulated earlier on. But we are able to start putting it together and understand the functionality. So, I think the potassium example that you used just a while ago is a classic one. There are a lot of farmers putting out a lot of fertilizer and actually still not probably having an impact with that system. And I get reminded by my cows or by nature all the time, and like I'm calving at the moment, and we've put a little bit of nutrition out where we put our pasture out, and I suspect, but we've had lots of rain, so it's a bit boggy getting out of the paddock, but I can see this little green strip that the cows haven't touched, and I suspect that it's where we had the belt slip on the spreader at one stage and poured a bit out. And time again, what I would have thought in the past was perhaps healthy plants and healthy crops just aren't, because the cows, it's the last bit in the paddock they'll eat, a bit like they won't eat around where their cow paddock's been. It's just out of whack and out of complexity because there's too much water soluble nutrition being taken up by the plant. So, and I know, we know that there's evidence out there that by oversupplying some of the nutrients we do in our sort of heavy input farming system is also shutting down a lot of this function of the microbiome in our soil, and we're delivering nutrients to the plant when they don't probably want them or in the wrong ratios. So it's about, the challenge now is it's not in our ancient soils about not applying some nutrition. It's about finding out what's not there, and it's about finding out potentially what biology is not active in our soil and why that is, and trying to bring that together. And I do feel that, with some of these emerging technologies and greater understanding, that we will be able to improve our productivity quite considerably over the next sort of 5 to 10 years as we embrace some of these new technologies. But we'll not only be increasing our productivity, we'll be producing something that is more balanced, whether it's more balanced nutrient density in our grain or in the pastures that we produce. So yeah, look, we've still got a lot to learn, but we can't learn unless we start testing and start trying to interpret the data. And it's really important for Peter to communicate with the landholder and track, the observational sciencing with some of the other stuff as well. And hopefully some of that observation can lead back into our, institutions and research institutions to actually do some more local research in our own context in Australia as well.
Yeah, agreed there, Stuart. On that, it was really important you talk about what the plant is missing and what we might be giving too much to, and we get very sold down the river in ag on putting something out and seeing an immediate response, especially growth. Hence the reason ureas and nitrogens are so popular, because you will put a fertilizer out and the plant has no choice but to take it in and have a response to that, which is usually a growth response. And yield seems to be king, unfortunately. But where we get sold again a bit down the river is understanding that we're not losing yield at the expense of doing this to our soil. We're actually usually improving it. An example I can give is we've been working with McCain's down in Tasmania for the last couple of years now, and they're pushing to 2030 for a full regenerative program for all of their potatoes that are grown and sold around the world. And we started with 3 growers in the first year just to get a bit of an understanding. They wanted to sort of test us and see where we were at, and we did all our DNA testing and we developed a biological program that was bespoke to what those customers were missing. And we found in the 3 different trials in that year, we had between 3 and 8 tonne yield improvements to the hectare, which they were very, very pleased with, which put money back in their pocket. But what really fascinated me was not only was there less scab on the potatoes, the ones that went in, 2 of the 3 customers went into storage and the potatoes stored longer with less blemishes. And those farmers then the year after they harvested their potato crop were growing steers. And one of the customers that we went to this year in February for our field day down there highlighted the fact that he got his cows to 200 kilos. He was 200 kilos heavier at time of sale on the pastures that he had put the biological program out and done the testing on versus where he'd gone a conventional program on the other side of the farm. And we see this regularly, whether it's beef cattle, dairy cattle with improved quality of milk. We don't necessarily see quantity improvements, but we see improved butterfat, improved protein, improved quality of milk. Same across the board with wine grapes, with olives. It's a consistent result we see over a period of a couple of years, and usually quite rapidly, by making those adjustments to allow that soil microbiome to be a bit more freed up. I think we go back to the original question, was planting multi-species crops. And the beauty of where planting multi-species crops is that each of those different plants require different things. So the idea around multi-species planting is to get a plant exudating and calling in different sets of microbes to create a bigger, overall, more diverse microbiome in the soil structure. So I would always say anything that's multi-species over monocrop is going to give you a better result over a period of time. And I think the person who asked the question asked about adding in certain microbes. This comes down to costs and timeframes. We can utilize nature to do our bidding. It just takes time. I can see a lot of the time we have these programs where people don't go and do a lot of inputs. Things will slowly improve over time and things will slowly come back. A lot of the issue we have in agricultural soils is that we've used chemistry and synthetic fertilizers over periods of time that have actually destroyed those microbes in the system. They're no longer there. So to get them back into the system and to turbocharge what you're trying to do, adding in these particular species back into that soil structure can extremely fast track what you're trying to do. And this is where we try to come in on the understanding of how these microbes behave, what type of environment they require, and if they're not being used, are they going to survive? You probably see a lot of biological products using bacillus. The reason we use bacillus so often is that they're the only species of bacteria that we've discovered that have the ability to go into what's called an endospore when they're not being utilized, or they can no longer survive and function around the rhizosphere. They will physically, as a colony, takes about 6 hours. They'll build a shell around themselves, and that shell becomes pretty much impervious, and they'll drop away from that root system and sit in the subsoils. Now, I don't know how many guys keep an eye on science and things going on with space science and stuff like that, or cores they dig out of Antarctica. But there's been a number of stories in the last couple of years highlighting the fact there was a core dug out of Antarctica in the last couple of years where they went down to 10,000 years ago in the ice cores and managed to revive bacillus bacteria in those ice cores with some on an agar dish with a food source. So 10,000-year-old bacteria that has just come back to life for the right conditions were available for them. They found a new microbe that was living on the outside of the International Space Station that we haven't even been able to name yet. They thought we discovered life on Mars when we first took the rover over there, but it was bacteria that had hitched a ride on the trip. They are far more hearty and more important than we ever believe. And if we look at even ourselves and the planet around us, I mean, every 10 cells in the human body, 9 of them are bacteria. We're only actually 1 out of every 10% human, if you want to put it that way. And that they're critical to our function on how we function. And biology is critical to how everything is pulled together. And as Stuart said, we're just really scratching the surface on it. But it's doing projects like this and looking at what's going on in that microbiome over set periods of time and being able to do these tests, analyze them, and then understand what impact the system has had on the system based on what you've put out, what fertilizer you've used, what chemicals you've used, the season you've had, how much rain there's been, whether we're in drought. All of these things build this dossier for your patch of land. That's why I believe it's so important that you guys are the ones doing these tests on your farms.
I was just going to say, I think it actually supports.
Sorry, lost you there.
Is that somebody trying to ask a question or just a mute fumble?
maybe a mute fumble.
If you do want to ask a question, yeah, feel free to jump in. Maybe put your hand up so we know that you are asking a question.
I might just answer Zachariah's question on phosphorus fixation. So, look, I've probably got pretty sandy soils here, and since I've been sort of working on focusing on sort of stimulating the microbiome, I really do struggle actually to get P responses, even at high yielding years. Now, that's not sustainable because obviously we're drawing down our P bank. But I think, yeah, like anecdotally, there's no doubt that, not anecdotally, a lot of the trial work that I've done at my own expense sort of demonstrates that with particularly N and P, that we're flattening out that response curve where we would have seen a response to higher rates in the past. And that has obviously huge ramifications as far as profit margins going, gross margin going. But yeah, that's one of my problems is that when I do some trial work, quite often the zero fertilizer treatments are just as profitable or close to being as profitable as some of the fertilizer rates. But we need to be mindful that we need to be putting in what we're taking out as well. But I think it's what form we apply our fertilizer in. And I think if it's too water soluble, then there's no doubt that we're shutting down some of that microbiome from working straight away. So again, I see that's where there's potentially a lot of work to be done on sort of trying to find that sweet spot where we're, getting some energy into our plants, when the biology might not be that active, where it's cold, wet, but we're allowing, we're taking account of when the biology starts to cycle and pick up, that we're not sort of oversupplying water-soluble nutrients in a way that sort of is not allowing the plants to function like they would normally function and pump exudates into the soil. I think, fun fact, a 3-ton cereal wheat crop could or should, and I doubt whether anyone, including me, is doing it, exude up to 4 to 5 ton of sugars into the soil. If you think about that, when we're putting other amendments on the soil, that's a lot of exudates that are going into the soil and feeding that microbiome. And that obviously takes energy. So there's got to be a good return on that investment. But I think we would agree that we've been drawing down on that organic carbon levels in our soil. And, really, if we start focusing on the microbiome again, we can create greater nutrient efficiencies, but hopefully we can start building that soil fertility and soil carbon and structure back in our soils, where we're not only creating more exchange sites to hold nutrition and cycle nutrition, but we're also holding more water in our soils. We know that sort of carbon can hold 7 to 8 times its volume in water, and in an environment that has, wet periods, not as often as we'd like, but a lot of dry periods, then, holding more water in our soils is something that we can do to build resilience when we're seeing greater sort of fluctuation in intensity and heat with our climate. So there's some really good reasons for building our soil health as well.
On that one too, Stuart, really quite interesting. We do a fair bit of pasture. We're based, just so you all know, we're based here in Victoria. We're on the other side of the country, down in a place just on the edge of Gippsland. And we have a lot of pasture customers down through here. It's just grown exponentially because of what we've been able to do for these guys and give them the right data and get them the right results. But we're constantly looking to see how we can improve systems. I mean, obviously for dairy farms, they're needing grass and pasture all the way through the year. And one thing they'll do through winter here, can get bitterly cold down here in Melbourne and through Gippsland in winter. So everything really shuts down significantly. And they'll put out a product called gibberellic acid, which will stimulate the plant to split cell or elongate their cells internally. Uses a fair bit of nitrogen to do that. One of the negatives you can get, or a couple of the negatives, it can really affect the chlorophyll production within the plant for the first couple of weeks after application. So everything yellows off significantly. And it can take a lot of energy reserves to do this. So at the beginning of spring, these plants can then have a couple of weeks of really poor growth coming out of this process because they've lost a lot of energy that they needed at that start of the spring growth. So what we've done, we've sourced an amazing company we work with down here with a product called Crop BioLife, and we add that in with the gib acid instead of the surfactant, and it's got a powerful surfactant in it anyway. And we put a little bit of liquid nitrogen to offset the nitrogen requirements. But what Crop BioLife does is it stimulates, actually an organic product as well. It's made out of flavonoids from orange peel. There's some weird and wonderful things out there on the market. And this particular product allows the plant to grow more stomata on the leaf, which is the openings where they respire and take in carbon, breathe out oxygen, and open those cells that open and close those openings by up to 50% wider for only a period of 2 to 4 weeks. So what this does, this really builds the plant's energy reserves during this process. It offsets the yellowing from the cell elongation, and it offsets any energy loss that they may get at the start of the spring season. And this was all developed based on these DNA tests on what was physically happening in the soil after we'd put gibberellic acid out, and seeing what was happening to the soil microbiome and how the plant had performed and responded to that. So again, it all comes down to doing that deeper dive, and this is where we advocate all the way through, is that we're mostly all going to go do something. We're going to buy something, put a product out, whatever it is, and make a decision based on the data that you've collected, or whether you think it's the time of year to do it, or your experience in farming. By collecting this data constantly and building this dossier for your farm, you can really get a far better understanding on what works and what doesn't work. And that's one thing we really try to work on with people, is understand that it might even not necessarily be the product. The product may be a great product, but it's been used at the wrong time or in the wrong instance. And therefore you did not necessarily get the response or the result you were looking for from that particular product, even though your neighbor did. This is the crazy thing about soil and microbes, that you can do the same thing that your neighbor did and he had a wildly fantastic result, and you can get a very poor result because your soil is completely different based on the way he has particularly farmed and the way you farmed over the last 5, 10, 15 or 20 years. So all of these things have to come back again. It's like going to the doctor and saying, well, my brother's sick, can I get the same treatment? It doesn't sort of work that way. You might be from the same parents and you might have similar DNA, but you have had a different life. You may be younger, you may be older, you may be drinking, you may be smoking, so you've got to work out what path you need to take for your particular treatment plan when you're going to a doctor. And I always laugh at I just regurgitate things people have said to me because they stick in my mind. And it's like I had someone say to me, when we're not soil testing and doing these things, it's like going to the doctor, sitting in his chair and asking for a pill, give me something. Well, the doctor's sitting there going, well, I don't know what's wrong with you. And you're sitting there going, well, it doesn't matter. Don't worry about the test. Just give me something. Give me something that I'm not feeling too well, and these are my symptoms, so I want something for that. And unfortunately, a lot of us farm like that. We farm without that deeper knowledge and understanding of what's actually physically going on in that soil structure, and making these decisions. And I tell you, I get frustrated with farmers that spend money and be the guinea pigs, especially for products. New fandangled products hit the market, or a new chemistry technology, or a great new fertilizer. The sales guy comes out, gets you all juiced up, you buy it, and you go, that didn't really do a hell of a lot. But it's constantly coming out of your pocket. And there's only a finite amount of money that you can spend every year. It's all about yield return. You've got to get that return on the investment you've put in to keep things going and tracking. And the beauty of doing it this way is that you can actually track and physically track your farm growing. You can physically track the soil structure improving, and you'll see those benefits in the yield data. Unfortunately, we seem to get sold down the river a little bit on that quick fix, the nitrogen hit, the chemical to get rid of that particular bug or pest. But the damage that does, even ever so slightly, to your system, and you continue to do that over and over and over and over and over again, that's when we start running into trouble, and we start to see that 100 kilos of urea didn't work this year, so I've got to go to 150, and then eventually you're on 200, and then you're on 300, and the same with super, same with potassium. All of these things need more and more and more added into the system, because now the system is becoming less functional and less efficient. So the things you need to do to the system become your problem out of your hip pocket. And the beauty with doing it this way is we could actually improve that system and reduce your costs, and improve the impact of when you do something, and get a far better response from that system based on what it needs, what it requires, what it's lacking, or what it's requiring at that particular time in that particular season. So I hope that helps as well.
Thanks, Pete. I'm wondering if you could address Paul's question in the chat as well about standard sampling depths and standardizing carbon assessments.
So we sampling depths are usually, again, it's a little crop dependent, but usually most crops, we're wanting to take the soil around as the root systems. We want to see what's going on in that root structure. So for instance, pastures around sort of 15 to 20 centimeters into the soil. Most, again, fruit tree crops are similar, around that drip zone in the tree. So we're getting it from the area where the engine is in the soil for the plant or the tree. But usually around that sort of 15 to 20 centimeters. Carbon is something that the further we can get that into the soil and embed it as part of the soil structure, the more likelihood is you've got of retaining it. You've got, in carbon, it's like the old adage of oils ain't oils, as carbon ain't carbon. You've got different forms and variations of carbon in that soil structure. At the front end, your leaf litter, dead organisms, things that are easily converted into that organic matter. They're being utilised by everything in the system, all the way to the other end on something like biochar, which is something that will break down over 100 years. So it's part of the actual soil structure. And there's two other variations of carbon in between that. There's the four main sort of versions of carbon that are in that soil structure, and getting the right balance. So there's, I think we've had probably 10 or 15 years of changing methodologies and different understandings of how we want to measure carbon. I know there's a lot of companies running around sort of trying to get farmers onto 10, 15, 20, 50-year projects with it. I reckon probably you, Stuart, have a bit more of an insight into this as a farmer, and from that course you did, we learned a fair bit on that course about this. But yeah, carbon's one of those things that, again, for me, I think we need to understand it a little bit better, and the impact it has in the system, and what form of carbon you've got in your system. Over to you, Stuart. Sorry to put you on the spot there.
Oh, still learning, I think. But
yeah,
I think when you look at, I guess, ideally for structure, we want to be forming some of that longer term structural carbon in the soil for holding nutrients and whatever. But I think we shouldn't lose the point that actually carbon cycling, as with all the elements that are involved in it, so the breakdown of our crops is a really, really important way of applying nutrition to our microbiome and to our crops as a result of that as well. So it is, it's important to recognise that some of that carbon we do want to be cycling quite rapidly, because that's a better way of feeding our crops as well in a more complete form of fertilizer, I suppose, like we might get from applying the compost or some of the other stuff. So, but ultimately, we want to be building, we want to be encouraging our roots to go down deeper each year. So I know with what I was doing, I was seeing roots probably going down, 25 mil each year. And creating a greater rhizosphere. Some years that's not possible if it's not wet enough down there. But yeah, we want to be encouraging our plants to sort of go deeper into the soil, either to bring up nutrients that we might not be accessing now and cycling to the surface for some crops that aren't as deeper rooted. But again, I think it's taking a more holistic view of sort of how soils function. And I guess, for me, if we're not doing that in the context of how plants interact with the microbiome, then we're really not going to get where we need to get to. I think I've been really pissed off, if I can use that word, over the years with a lot of people that say, we'll never get our soil biology right until we get the physical and chemical property of our soils right. I argue strongly that you need to work on all three at once. But for me, the main one is the soil microbiome, because that's the bit that holds it all together. That's the bit that's going to keep changes that you make to your soil. I think any of the other interventions that we make are very, very short term. Deep ripping is a classical example in Western Australia where we get a response to deep ripping, and we started at 30 centimetres, and now there's people out there deep ripping to a metre every year, And, wow, that's expensive. Fuel's just gone through the roof. But, if we get an active microbiome, then we can make that happen. So, anyway.
It's that carbon nitrogen cycle too, Stuart, as well, the carbon nitrogen cycle. That's why it's listed on your soil test. It's critical. Microbes need both. And I don't know if people actually understand that when you put a nitrogen source in, especially urea or something that's very water soluble, that actually allows the microbial populations to explode in number. What they then need is carbon to offset that. So if they don't have it, or they don't have the forms of carbon they require, they'll find it and eventually start to drop away in numbers and die out. And we see this regularly on sandy vegetable production. Vegetable production is one of those classics, sort of, we're getting 4 to 6 crops a year. We're not really thinking a hell of a lot about the soil. We're just going to put everything in it we need to get that crop out, and we're going to put a lot of nitrogen down. And we regularly see carbon levels of 1 or less in the soil. And the microbes will find carbon out of organic content. That's their last bastion of it. So they'll start pulling carbon and degrading your organic matter in your soil as a survival mechanism. So understanding that these microbes aren't just your friends and allies, they are microbes work in different ways. Some of them want to protect the plant because their main food source comes from the plant's exudation. Pathogens will attack the plant and take everything they need. And then there's a whole bunch of microbes that live in that soil structure, which is probably over 80% of them that don't do either. They're just there. They're just there to function. They survive. They go under the radar. They've even discovered that pathogenic bacteria and fungi will act in a beneficial mode to the plant if they're overpopulated with beneficial microbes that will kill them, and vice versa. So it isn't just this hard and fast system where if I put something in here or I do that, I'm going to get this response. The response comes down to a raft of different mechanisms and things that are happening behind the scenes over a period of time that then how then the plant gets what it wants or it doesn't get what it wants, basically. So we've by testing this is the beauty of actually looking at the soil microbiome, because you can do something, you can think, I've done all these great things, I've put compost down, I've put all organic materials, I've stopped using as much synthetic fertilizer, I've cut my chemicals back, and things sometimes can go backwards. And you sit there scratching your noggin going, what the hell is going on here? I've done everything I've been told to do. But being able to look at then the pulls and pushes that are going on in that soil structure biologically then gives you more answers to the puzzle that you're trying to put together. It's like that. I always, again, look at it like a 1,000-piece puzzle, and 300 pieces of them, maybe 400 in the blue sky. And you don't have the box to tell you what the picture is anymore. You've just got all of these bits and pieces, and you're trying to put the pieces down on the page or on your desk to try and go, well, hang on, it's a sailboat. Now I know sort of what I'm doing here, and the rest of the pieces fit a bit better. And that's the collection of data. You know, it only comes down to when you get a few more pieces on the table that you're going to understand what that picture looks like. So the more pieces you can put together and the more testing and data you can collect over a period of time is far better than going out and buying a ton of urea. That's my take anyway, because that ton of urea is very short-lived. It'll get you a bit of a growth. It'll do maybe what you wanted it to do at that particular time, but then you've just kicked the can down the road for problems. That's all you've done. You've just kicked that can down the road. So that's me on that one.
All right. Thanks, Pete. I think, yeah, we'll have to leave it there for this morning. I know there's probably a couple of unanswered questions remaining in the chat, but I can chase those up and get some answers for you, and we'll send out a follow-up email to everyone with everyone's contact details. And yeah, we'll try and share some of the example reports from GreenMate. And yeah, we'll send a recording of the webinar as well, so you can rewatch it if you want to. So yeah, thank you everyone for coming again. There should be a slide on display with the Regen WA email and also a link to find more information about our natural capital accounting project that this webinar is funded through. And that one's, yeah, supported by the Western Australian State Government State NRM program. So yeah, thanks for coming again, everyone. Thank you, Stu and Pete and Ben, for sharing all of your information and experience. And yeah, feel free to shoot me an email if you want me to find any more answers or get you any more info, anyone who's on the call. Otherwise, yeah, there'll be plenty more opportunities to participate in our natural capital accounting project. More webinars, more events. We'll be sharing case studies, and the actual NCA reports will be publicly available. So yeah, you'll be able to get a bit more of an in-depth understanding this time around. Yeah, thanks everyone. Have a good Arvo.
Thank you, everyone.
Thanks
Appreciate you
for
coming.
Carrie, and for everyone attending.
All right, see ya.
Thank you. Bye.
See ya.